Um, so this is the first of 3 lectures I'm going to be giving. So this first one is entitled Becoming Human. We've had to slightly rearrange things just to get everything in place. So this one's going to be Becoming Human. And in this talk, I'm going to cover humanity's place in nature, who's our closest relative, and what have comparisons between human and ape genomes taught us about what it means to be human. We're going to look at evidence from genes and genomes. So I'm a microbiologist. I work on microbial genomes, but I do dabble in human genomes as well and looking at that kind of information. And that's where I'm coming from. The next lecture will be on the scatterlings of Africa. That will be on Friday. And then it'll be another couple of weeks after that. I'll be doing the third and final lecture of mine called Waking the Dead. And all of these will be looking at evolution from the view of molecules and looking at sequences. For some of you, I suspect that might be a little unsettling because you might not have done much biochemistry in the past, but I hope as well, that's the kind of way it works. I've also put up a chapter, a PDF of the proofs of a chapter from my book, The Rough Guide to Evolution, a chapter on human evolution. It was written in 2008, so it's a little bit out of date, but actually most of it is still basically true and reasonable. Okay, so that's just where, in terms of the support you're going to be getting. And if you want to interrupt me at any time with questions, feel free to do so. When we look at human evolution, we can look at various sources of data to look at the branch points and the changes that happened along various lineages. If we're looking at living primates, that gives us one view. We can then look at molecular phylogenetic and genomic studies. We can compare the morphology of humans and living primates as well and make inferences that way. So that's a useful source of information. We've got extinct hominins. These are organisms that are on the same lineage that has led to humans from our last branch point where we branched off from the most recent, our closest living relative. Here we've got the fossil record, and the fossil record can give us information about morphology. We can get some chemical information, and in fact now we can start to get ancient DNA perhaps. Certainly for one group, the Neanderthals, we have got that, and you're going to be finding out more about that in a later lecture. And then we've got anatomically modern humans, sometimes abbreviated as AMH, people like you and me. And we have evidence from modern day population genetic studies looking at mitochondrial DNA, Y chromosome, autosomal genes, and now looking at the whole human genome and comparing that with other organisms. So in this lecture, I'm going to look at 2 issues. One is evolutionary history, and the other one is evolutionary biology. So under the banner of evolutionary history, there's this question of how did we come to be what we are? How are we related to other organisms? How are we related to other species on the planet? When did we split from our closest relatives? When and where did our species originate? I'm not going to say so much about that point. In subsequent lectures, we're going to look at how did humans people the world, and also what was the relationship between our species and other species that might have been knocking around in times gone by. Those are questions about history, if you like. And then there are these questions about biology. What makes us us? What makes us human? What were those genetic changes that make us human? And what evolutionary processes work in our genomes? What adaptive changes have occurred since we became anatomically modern humans? And that's going to be in subsequent lectures rather than now. So where did we originate and who are our closest relatives? If we go back to Darwin, and Darwin is obviously the start of modern biology pretty much, for almost all aspects of biology. And in The Origin of Species, he actually didn't say much about human evolution. One throwaway line, which was that light will be thrown on the origin of man and his history. And if those of you are interested in reggae music, we did an Origin of Species in dub album a few years ago with a Jamaican guy, and that line comes up in the track there, one of the tracks we did on that. Light will be thrown on the origin of man and history. And there's huge amounts of fossil evidence that have come about since Darwin's time that has thrown light on the origin of man and his history. But obviously that's just one part of it. Looking at modern animals, what else can you say? Well, Charles Darwin himself speculated that humans originated in Africa. And the reason he said that was because, from this comment here, he said that basically the living mammals are closely related to the extinct ones, and that Africa was probably inhabited by extinct apes closely related to the gorilla and chimpanzee, and that these new species are now our closest allies. It's more probable that our ancestors, our progenitors, lived on the African continent and elsewhere. So, as in many things, Darwin was right and was prescient and ahead of his time. Occasionally he made mistakes, but most of the time he was spot on. Darwin's bulldog, Thomas Huxley, also took this further forward and wrote his book, Evidence of Man's Place in Nature, and said it's quite certain, this is based on morphological comparisons of skeletons and so forth, that the ape which most nearly approaches man in the totality of his organisation is either the chimpanzee or the gorilla. And so that was basically where things stood in terms of morphological comparisons, what you can tell by looking at skeletons and anatomy. And that basically was where things stayed for 100 years or so in terms of our understanding of things. But if you think about this, just look at the pictures. It's bloody obvious, isn't it? If you look at which are the sister taxa here, which two are more closely related, it's obvious to anyone, isn't it, that gorillas and chimpanzees are more closely related to each other than either of them is to us. So, okay, these, as Huxley said, these are maybe our closest relatives, but they're more closely related to each other than they are to us. And not just looking at the superficial, they're hairy apes, and we are these wonderful, hairless, beautiful creatures. Humans have many other adaptations, bipedalism, these giant brains, there's tools, culture, language, whereas these guys, they're knuckle walkers, and the ancestor must have been a knuckle walker, and we descended from something that was like that. And in that view of the world, the only doubt was that where did humans branch off? Did they have a really long ancestry and branch off before the orangs? So the orangs are even closer to these guys, or did they branch off after? Problem with looking at this kind of thing just from morphology and from the fossil record is that there are no chimp and gorilla fossils, which does hamper things somewhat. But that's basically the man on the street's view. That was the view that people would have taken up until perhaps the 1960s. In fact, there was some very early work in immunology in 1904 where it showed that if you take anti-human anti-serum, it reacts with chimp and gorilla serum more strongly than the orangs and gibbons, backing up using molecular approaches, if you like, to back up what you get from morphology. And then in the early 1960s, similar kind of immunological approaches and protein electrophoretic approaches basically confirmed that humans are effectively great apes. We're related to those great apes most closely than anything else. But it was in 1967 that the kind of revolution happened in our understanding from this paper from Sarich and Wilson, where they looked at immunological approaches using these kind of antibodies and looking at what bound most closely to what. And they found that basically that humans and apes must have had a common ancestor about 5 million years ago. If you take the view that humans and old world monkeys separated about 30 million, then it's about a sixth of the distance to the branch point between us and the great apes. And this was quite a revolutionary way of thinking, because people had assumed, they just assumed that humans must have had a very, very long distinct ancestry separate from the great apes, because we are so different, basically, in our mind, so superior, like some little Lord Fauntleroy, we've got that long ancestry, and it can't be that we share a relationship with a hairy ape only 5 million years ago. But it turns out that Sarich and Wilson are probably right. The next, around the same time, breakthrough in our thinking came from a paper in 1965 by Zuckerkandl and Pauling. Linus Pauling was a Nobel Prize winner. And he basically said, look, actually, you can look at sequences of molecules, like DNA sequences and protein sequences, and you can use those as documents of evolutionary history. So it's the first time the idea awoke in kind of consciousness that you could actually use sequences as an independent way of looking at evolutionary relationships and the timing of evolution separate from looking at pure morphology. And in the 60s, another guy, this guy here, Kimura, came up with a novel understanding of how sequences change, which he basically said that most of the changes that happen to sequences are neutral. They don't make any difference to biology at all. And some of you may remember that the genetic code has this redundancy, and that third position in the genetic code, it doesn't really matter. So if you've got a triplet like that codes for glycine GGG, if it changes to GGC, that doesn't make any difference to the biology because you still get a glycine put in the thing. But those kind of changes are still informative when it comes to working out evolutionary relationships. And so you can take DNA sequence data. This is a bit of an old slide because this is showing you what we used to have was chromatograms. It's all done differently nowadays. But you can line up, say, a bit of sequence here from a human, a chimp, a gorilla, an orangutan, and you can look at those sequences. And when you've aligned them, and you can look at where they agree with each other and where they disagree with each other. So at this position here, those three A's agree with each other. The orangutan's got a different one, so we can say, the orangutan's differently related to us. That's a separate group branch here. But here we've got one where the chimp actually is different from the others. So these so-called polymorphic sites are informative sites, phylogenetically informative sites, what we use to then draw trees. And we can draw trees like this where we can actually come up with branch points based on the sequence similarities. So let's go back to the relationship between humans and our most closely related living relative. What is it to be? So if you approach this with an open mind, you effectively we're left with a trichotomy because let's just say that it's not obvious that these two are more closely related. Let's just say we don't know. We end up with this trichotomy. Who is the closest relative here? Are gorillas and chimps more closely related to each other? Humans and gorillas or humans and chimps? Or is it really that they, and this is a thing that evolutionary biologists really hate, is it really that there was a split at one time where three different paths diverge? They always like it just to be bifurcations, but maybe it wasn't. Maybe it was a trifurcation at this point. So the earlier molecular phylogenetics, trying to use these sequence-based approaches in the 70s and 80s, they were very clear that yes, humans, chimps and gorillas belong together in what we call a clade in this branch. But the sequences were all very similar. And from these early analyses, just one amino acid change supported the idea of there being a human-chimp clade, for example. People used other approaches, so kind of indirect approaches. So if you take DNA and you take, say, human DNA and chimp DNA, you melt the DNA. So you basically zip, it's like a zipper. You unzip the DNA in both samples, and then you mix them together. If they are closely related, then they zip back much more quickly than if they're distantly related. And this can be measured by measuring this melting temperature. And studies on that, using that kind of approach, found that maybe the human-chimp complex was more stable than the gorilla-chimp or the human gorilla. As time went on, as we got into the 1990s, still getting contradictory results if you looked at one gene at a time. Some genes would say gorilla was the closest relative of the chimp or closest relative of humans. Sometimes it was the human and chimp went together. But as you got to the late 90s, it was clear when you added all the data together, it started to become much, much more supportive of the idea of a homo-pan clade. So that's humans and chimps together. And only a few that support other relationships there. And so you might say, well, why isn't it clear-cut? Shouldn't every sequence that you look at give the same answer? And it should be clear, shouldn't it? Well, the reason that it isn't that clear is what happens sometimes is you get, within a population, you get a split. So let's say this gene here is behaving in a tidy way, the dotted line one. So this population here splits off, this one splits off from that one, and the genes march off in a sensible order. If you read the sequences out from those genes, they would allow you to recapitulate that pattern of branching. Whereas this one in the darker colour here, this one is actually, there's a split within the population. Let's say everyone originally had brown eyes, and then there's a group that got blue eyes that split off here, and another group here that stayed brown-eyed. If you looked at the way that that's segregated, it may be that in one population, just by random chance, this particular lineage died out in that direction, but survived in that direction. And when you start drawing trees, you end up with trees that don't equal the species tree, and give you a misleading result. Now, this doesn't happen all that much, but it does happen enough to just create some noise. But if you take the consensus from many thousands of genes, you can actually come up with an answer. Here's just before we got to the stage of sequencing genomes, there was one important paper here which looked at what we call ALU elements. So these are repetitive pieces of DNA in the human genome which can jump around. But they don't jump around very often. So if an ALU element's sitting in a piece of the DNA and it jumps to another point of the DNA, you can look in the pattern of those insertions on each lineage and work out the branching pattern. And that's what they did here, and they basically came up with a very strong support for the idea that humans and chimpanzees were more closely related to each other than they were to the gorilla. And then these were all more closely related to each other than they were to the orangutan. In fact, around the same time, people started looking at, with an open eye, again, with a completely open mind, at the morphology. And even there, from morphology, they could come up with some evidence for a human-chimpanzee clade there. There was one rather contrary paper here that suggested that humans were most closely related to orangutans on basis of morphology. I think that's now been completely disregarded, but certainly there's evidence from morphology that there was a human chimp clade. So before we got to the stage of sequencing genomes entirely, we had all these different lines of evidence coming together, and this is taken from one of the reviews I put in the Dropbox there, so that early on we couldn't resolve this trichotomy. Chromosomes, I haven't said anything about that, but chromosomes suggested humans and chimps went together. DNA hybridization, supportive of that. Sequences, supportive of that. In fact, other evidence, gene expression profiles, and then morphology as well. So we're now reached a situation where we have resolved this. And we've resolved it even more so now because in the last few years, we've now got genome sequences for all of the great apes. And in fact, some of their relatives as well. So we've got macaque genomes as well to help us triangulate things. So when we put all this evidence together, it now becomes quite clear that we are more closely related to the chimp than we are to the gorilla. But counterintuitively, the chimp is more closely related to us than it is to the gorilla. So that's it's kind of a fairly mind-boggling thought, I think, to think that we are really that much more closely related to the chimp than the chimp is to the gorilla. And the evidence that superficially bound them together has actually been questioned. And you are going to get some lectures on this kind of thing later. In terms of knuckle walking, it was thought that these two both knuckle walk, and therefore the ancestor of all three species must have been a knuckle walker as well. But it turns out that first of all, gorillas don't knuckle walk as much as you might think, but also they seem to chimps and gorillas have evolved knuckle walking as a derived characteristic independently. And there's no evidence that the most recent common ancestor, was a knuckle walker. So it's kind of, and you're going to have some lectures about how, in fact, what's called arboreal bipedality, where actually standing upright and walking in the trees, even goes back as far as the common ancestor with orangutans. So the assumptions had to be thrown away. In fact, there's been also some molecular studies done on bonobos. So one of the interesting developments in the last 50 years is we realised actually there are two different types of chimpanzee. There's the common chimpanzee, but there's what used to be called the pygmy chimpanzee. I think politically correctly you now call it the bonobo because it sounds a bit rude to call it a pygmy chimpanzee. And they are really quite different from common chimpanzees. So a lot of people, there's a lot of what you might call just so stories when you look at human evolution, and you look at what chimps do, and that tells us why humans are like we are. You know, chimps engage in genocide and war, and they hunt monkeys for meat, and it's a male-dominated society. But in fact, bonobos, they're the opposite. They're the kind of hippie society. The females dominate, and the currency is sex, basically. Everyone just gets along by having sex. And it's just a little joke here. You know, basically, they're trying to film bonobos. When people were looking at bonobos in zoos, in fact, they find, you know, it's quite shocking watching what they're getting up to in terms of sexuality. Here's a couple of female bonobos engaging in lesbian sex. They're basically doing genital rubbing. So they have sex with each other, heterosexual and homosexual, all sorts of things going on there. And their genome's been sequenced as well. Now, of course, this is a bit of an old slide because it goes back to the days of George W. Bush, but maybe some people would say you didn't have to go and do all that molecular sequencing and genome sequencing to work out that some humans are clearly very closely related to chimpanzees. But there we are. Now, more recently, we've had the gorilla genome, and the point about this is that once you've got multiple genomes, you can then start to kind of triangulate where things sit in terms of their branching patterns. And we can see that actually a very, very little difference between the human-chimp divergence and the human-gorilla divergence. With chimps, it's about 1.37% change in sequences. It's 1.75 for gorillas. So the actual separation time between when we separated from the common ancestor with chimps and from the common ancestor with gorillas, it was quite a short period of time. One of the problems that we have at the moment is that people aren't quite certain when this divergence took place. And if you try and work out, these are various evolutionary models based on mutation rates as to when the human chimpanzee ancestor was, it is still roughly thought to be about 5 to 6 million years ago. But there are some fossils that have been found in recent decades, Orerin and Sahelanthropus, which sit at that time when we were supposed to be diverging from chimps, but look more human-like than you'd expect if they were the ancestor. And there's been you can read in my rough guide to evolution, there's a lot of discussion about what that all means and whether it's true or not. When you look at the bonobo genome, you get the same branching pattern. Humans are clearly related to the chimps, but we're not more closely related to either one of those two species because they branched off after us. But 95% of the sequences give you one tree with bonobos and chimps here and then humans as the outgroup. But a small fraction, because of that what we call lineage sorting issue, you get bonobos and chimps. We got where there's a longer time period to the ancestor of bonobos and chimps, and here sometimes the bonobo and human are actually more closely related than the bonobos and chimps, and so you can see that there is these discrepancies for occasional genes, but when you take the consensus of the whole genome, you end up with a clear picture, which is that we separated off from the chimps. In this one, they even went as close as 4.5 million years ago. And then these two species separated out about 1 million years ago. Now, one of the strange things when people compared the human and chimp genomes was that if you look along the position across the genome, you get a certain idea of how much variation there is, how much mutation there's been, and it's been fairly much the same for all of the different chromosomes here, apart from the the X chromosome shows very much different level of divergence, a much lower level of divergence than you'd expect from looking at all the others. And one of the early papers, this paper from Patterson et al. In Nature, said that maybe what that meant was that there was an initial separation 6.3 million years ago, and then one of the lineages from the human lineage actually then rehybridized with chimpanzees, and there was some exchange of genetic material, and therefore it wasn't a simple split. There was a split and then some re-injection of material from that lineage. In fact, if you look, as I did this morning, just checking up on this, in response to that paper in Nature, there are other people who have said, well, that may not be the right explanation. It may be that there is actually a lot of selection for genetic change on the X chromosome, and the genes on the X chromosome have been kept that way because of they're so important and such like. And like many things, you know, there's a lot of controversy about these kind of findings, but this was an interesting early interpretation of the molecular data. Now, it's worth pointing out that we are not 6 million years separated from a chimp. So, you know, if it's 4.5, 5 million, 6 million, it's not that we are 6 million years away from a chimp, because that is a false view of how evolution works. Because basically, we're 12 million years away from a chimp. We're 6 million years away from the last common ancestor with the chimp. But don't forget, the chimp has been evolving in its own way. That lineage has been evolving in its own way. And again, it's one of those easy to jump to conclusions. Oh, the ancestor must have been like a hairy ape, like a chimp. Well, how come? Maybe it was more human-like. And the chimps have diverged away from the human-like ancestor. And there was an interesting paper that came out a few years ago looking at this ancient hominid Ardipithecus ramidus from about 4.5 million years ago. So somewhere along the human line, but not far from the most recent common ancestor. And basically, looking at the morphology there, even that far back, it looked as if this human ancestor or close human relative was actually more human-like than ape-like. So as it says, lacks any characters typical of suspension, vertical climbing, or knuckle walking. And despite the genetic similarities of humans and chimpanzees, the ancestor probably differed substantially from any extent African ape. So our ancestor with the chimps did not just look like a chimp. And there's a video from Owen Lovejoy, who actually described Ardipithecus on YouTube. It's only 5 minutes long, but he's describing his interpretation of human evolution based on that. Okay, so that's the first half of the lecture. It's going to take maybe slightly more than half the time, but we'll have to go a bit faster. The next question to look at is, what is it that actually makes us human? So what actually happened along this branch here, when we did split off from our most closely related extant ape species? What happened along here to make us look like this rather than like that? And there are two ways you can look at this. You can look at it gene by gene, and some of that has gone on. But you can put your foot on the accelerator and look at the whole genome and compare the whole genome of living animals with that of humans, living apes, and look for differences in the genome. But when we look at humans and our closest relative, the chimpanzee, we've got 35 million base pairs of difference. So actually working out what is it that makes us human and chimps, it's going to be a tractable problem in that it's finite, in that we know that difference is there in the differences in the genome, but actually working out what are the significant things is going to be a job of work. There's going to be no easy answer. And most of the changes that we see, if we looked at your genome and a chimpanzee genome, would be neutral. They would have no effect on biology. Like I mentioned earlier, that changes in the third position of a codon. They don't make any difference. And what we call adaptive changes that actually make a difference to whether you survive in your particular niche and whatever, are going to be a small fraction. And it's clear that there's no simple linear relationship between morphological changes and sequence variation. So the morphological differences between a human and chimp are much, much greater than they are between two mouse species that might differ by the same amount of DNA as we do in chimp. So there's a lot of change going on in morphology, but it may be only small amounts of change in the DNA. And by comparison, if you take dogs, all the difference between a Chihuahua and a Great Dane is actually put into a very small fraction of a percent in terms of DNA changes. So there's no simple one-to-one relationship between a certain degree of morphological change or behavioural change and sequence change. And there are various kinds of changes going on in the genomes. You can have changes in the amino acids. Sometimes those will have effects on the function of the protein. Sometimes they'll be neutral as well, because sometimes one amino acid is pretty much the same as another amino acid. You've got deletions of exons. You've got large and small indels. So the indel is a shorthand for insertion or deletion. It's our way of saying that we're agnostic about whether a thing was shoved in there or something was pulled out when you compare two genomes. Duplications, got substitutions in promoter regions. So it may be that you've got the same genes, but they're switched on in different ways. And that's all that makes the difference between humans and chimps. That could be. We don't know. All of these things could be playing out during evolution. Now, we know when we look grossly at chromosome structure, there are differences. And the biggest difference is that we have a different number of chromosomes. So our human chromosome 2 is in fact a fusion of two ancestral chromosomes, which are reflected as chimpanzees' chromosomes 2A and 2B. And I think there's similar patterns of chromosome distribution in gorillas and orangs as well. So this is something that happened in human evolution, that those two chromosomes fused together. There are segmental duplications, inversions, and translocations. Bits of chromosome move around. So this is all a very gross level. If we look at single nucleotide substitutions, we come up with a figure of about 1.23%, which is a very small amount. This idea that we're about a percent away from chimpanzees is not far wrong. If we look at proteins that we say are orthologous, same protein, you take your hemoglobin protein or whatever, myoglobin, it will differ by just two amino acids. And coming back to that earlier point, it doesn't mean that those two amino acids have accumulated in our lineage. One change might have accumulated on the chimp lineage, one in the human lineage from the ancestor. And nearly a third, 29% of proteins in you are identical to those in the chimpanzee. There are differences in our repertoire of endogenous retroviruses. So retroviruses can jump into the genome and lodge in the genome as we get infected with them and they move around, and they can then lose the ability to come out the genome and just get stuck in the genome. And we have different repertoires of those in humans and in chimps. And also I mentioned about allele repeats as well. There's more allele repeat jumping around in the human lineage, it appears, than in the chimpanzee one. In addition to the observation, the X chromosome seems to be more closely related between chimps and humans than you might expect. The Y chromosome seems to have undergone a lot of changes in the evolution from humans and chimps, and it's not quite clear what that means. Why is that, and what does it mean? But it's an observation that's there. There are clearly some differences in biology between humans and chimps. So in terms of apoptosis, one of the caspases, caspase 12, is disrupted in humans, but it is intact in mice and chimps. And one of the things that we get that they don't seem to get is Alzheimer's. And people have speculated that it may be that this is quite a contributory factor there, although we really don't know. There are changes in the inflammatory repertoire of inflammatory signaling molecules, parasite resistance. There's a particular change in the sialic acid biology. So these are molecules that get coated onto the surface of cells. So the coating that our cells have is subtly different because of changes in this particular CMAH gene in the human lineage. One of the weird things that was discovered, counterintuitive, was when the genome of the chimpanzee became available and you looked at comparison across the whole genome, particularly in genes that we know in humans are associated with genetic diseases, sometimes what we found in the chimpanzee was the allele that looked like the disease state in humans. So here's a number of variants that have been described in human populations as being associated with a disease. Maybe not, the association is not one to one. It may be that there's increased risk, say, of insulin resistance with that particular allele. But in the ancestral state, sometimes you might have, sometimes in the chimp you might find a disease state, and you might actually be able to work out that the ancestral state was what looks like disease today. So what must have been happening in evolution is that changes in other proteins, in the signaling pathway or whatever, have also occurred in parallel, so that what used to be a disease state becomes the wild type state, and vice versa, in ways that don't involve just changes in one protein. Now, looking at the comparisons with humans and chimps, and more widely, there is now a long list, and this is only a short abstract of that long list. I've put in a review which comes from last year, which is, as far as I can see, is the most accessible, up-to-date review of these changes. But we don't have time to go through all of them in detail, so I'm just going to quickly look at two genes in more detail before we finish. So one gene that's been a focus of a lot of interest is a gene called FOXP2. And this was first identified in humans as a problem in genetics because there was a dominant mutation passed on in a Mendelian fashion in a family, human family, that's been given the name KE, the KE family in the literature. And this family had a problem with speech. And there was a missense mutation in this protein. So this is a regulatory protein, and it has a particular domain in it, the forkhead domain, and there was a missense mutation that turned an arginine into a histidine in that particular domain. And it regulates hundreds of genes in the brain, and it's then been dubbed the language gene because it's one of the few genes where it clearly has an effect on language. So it's kind of a bit of a mystery that one of the things that humans do is that they acquire language, and they do this very early in life. You can take a chimp and you can take a human, you can bring them up the same way as the human starts speaking, the chimp doesn't. And so there must be some genetic basis and neurological basis for our ability to acquire language, but it's fairly opaque at the moment. We don't really understand. And this was one of the few clues where we got a gene that actually did have an effect on language. And it appeared that this gene had undergone evolution in bird song and probably in bat echolocation as well. So it's kind of intriguing there. But the weird thing was that it turns out that if you look at this, the sequence of this gene, or particularly of the protein, and you plot out changes between, say, mice and primates, and then within various primates, what you find is that we've got, if we look at neutral mutations, we've got 131 that separate us from the mouse. We've just got one actual change in the amino acid sequence on the lineage that separates the mouse from the primates. And then among the primates, there's very few. There's none from the ancestor in the rhesus, and none in this pathway, one in the orang. But then here, go along here, and suddenly in humans, we've got two changes to the amino acid sequence, and none in the that don't change the amino acid sequence since our ancestor from the chimp. So this makes us think that this must be a gene that's undergone evolution on the human lineage and has some functional role in what makes us human. That's a story that is being told at the moment, and it's quite intriguing. So what people have done is they've made knockouts in mice, and they show that this gene is essential. If you knock out both copies of it, you get a severe developmental delay, and actually they die. And then what you can do is you can knock into the mice the the mutation that affects the human populations, this human family that gives them trouble with language, and they get developmental difficulties as well, but not quite as bad as knocking the whole gene out. And the weird thing that they've done is that they actually took the human gene, the sequences in the human gene, and they put that into mice. And the mice, generally healthy, they have slightly different vocalizations. They don't start speaking. I mean, some of the cartoons produced around the time was like, the mouse gets up, it's like Mickey Mouse saying, "Hi, what's up, doc?" kind of thing. Not quite that, but they do show changes. And so this is giving us a glimpse of how we're actually going to be able to sort these questions out of what it is that makes us human. How did these changes that occurred in our lineage since the chimpanzee play out in terms of changes in behaviour and morphology and so forth. One other gene I'll briefly bring your attention to because I want to play a video before we finish. Have you got another lecture after this? We absolutely have to finish by 10:02 then, don't we? All right, so amylase, salivary amylase in saliva, breaks down starch. And there are multiple copies in mammalian genomes, but there are many more copies in the human lineage. So there's been an expansion of the number of copies of amylase, the amylase gene, salivary amylase gene. And this correlates with the production of amylase. So the more copies of the gene you have, the more amylase you produce in your saliva. And people who have high starch diets have more copies of AMY1. And so the assumption, I put a recent review arguing this, is that actually this amylase is an adaptation on the human line to eating starchy vegetables, but particularly cooking them. And there's a book even being come out called Catching Fire, How Cooking Made Us Human, that's basically being able to eat things like potatoes and whatever. You have to cook them first, and it liberates so much more starch if you cook them, and then the amylase has evolved as a way of handling that starch. And the interesting thing about this amylase in the saliva is it breaks down the starch immediately, and particularly in infants, this means that you get immediate access to glucose from your diet. And so this may give you access to more calories more quickly. There have been some suggestions that multiple copies of amylase make you fat, correlated with obesity. That doesn't seem to be true. There's a more recent paper that says that's probably not true. But there is this. Particularly if you've got diarrhea or your GI upset and you're not coping, being able to get to absorb glucose directly in the mouth is actually obviously an adaptation. So where is all this going? Basically, we can look at all of these different primate species, and we can tease apart the changes that we see in the genomes. And now we have these amazing methods you may have heard of, CRISPR-Cas9 genome editing. So we can go in and make changes. So we could take chimpanzee cells and we can make them human in a particular locus, or we can take human cells and make them chimpanzee-like, and we can then go on and look at how they differentiate. We can even go and perhaps get to the stage of growing organs and whatever. We can do all sorts of analysis on this. So what I think will happen in your lifetime, in your working lifetime, we are really going to unpick all this, and we will come to know what it is that has made us human, what actually has changed since our ancestor from the chimpanzee that makes humans special. Of course, one can't really talk about this subject without also pointing out that the future is not necessarily bright in terms of our ancestors, our most recent our closest relatives. And in the chimpanzee genome paper, they point out that there are so few differences between us and chimps. Maybe we should take more care of the chimps and of the great apes. And the number of humans now is over 7 billion. These are all under threat. There used to be over 1 million chimpanzees at the beginning of the 20th century. We're now down to 100,000. And there are those who say that we should be giving them equality and have been court cases and such like. And it's an interesting question as to how far you push this. But it does underline the fact that there's so little that separates us, that we do have an obligation to keep these populations going. Now, what I'm trying to do in my 3 lectures, I've rambled on a bit, you're starting to get a bit bored. I always like to put a bit of entertainment in. And so what I've just got here is a video which will summarise what I've said and also lay the foundations for the talks that are to come. And because I never trust the microphone to plug into the system, I mean to plug my computer in, I'm just going to put my microphone next to the computer and off we go. Oh, wait a minute. No, we don't have to click on it and then it will work.
Man is a singular creature.
There we go. I'll have to come out of PowerPoint. I don't know why it's not playing in PowerPoint, but we can go in here and just play it from here then. And in fact, I put it on. There we go. I've also put this available for you on Dropbox if you want to watch it. It's also on YouTube. And in fact, the Symphony of Science.
Man is a singular creature. He has certain gifts which make him unique among the animals, so that, unlike them, he is not a figure in the landscape. He is the shape of the landscape.
Another Birmingham person.
Of Africa.
Yeah, help the print.
Of Africa. We say this is what we can. This is what we can.
In a parched African landscape, man first put his foot to the ground, to the ground, to the ground, to the ground.
Africa was our only home for tens of thousands of years, until a small handful of people made their way. Ocean of Africa. These beings with soaring imagination, eventually found themselves and their machines into interplanetary space. These beings with soaring imagination, eventually found themselves and their machines into interplanetary space. We are children of Africa. Children of Africa. This landscape has been home to humans 200,000 years. We have come so far. All of this is cause for great celebration.
We always laugh at this.
It's a story about us. Those early Europeans were people like you and me. But it is humbling when you see the challenges they faced. People like you and me overcame the Neanderthals. People like you and me made it through the Ice Age. These beings with soaring imagination eventually flung themselves and their machines into interplanetary space. We are not the only beings with personalities, minds and feelings. Chimpanzees have very clear personalities, minds and feelings.
Human-chimp brain fetal analytical with 3 more rounds of division to help cook symphonies and ideology.
We are not the only beings.
Everything that we are that distinguishes us from chimps emerges from that 1% difference in DNA.
Like you and me overcame the Neanderthals. Like you and me made it through the Ice Age. These beings with soaring imagination eventually flung themselves and their machines into interplanetary space. These beings with soaring imagination eventually flung themselves and their machines into interplanetary space.
Using his burgeoning intelligence, this most successful of all mammals has exploited the environment to produce food for an ever-increasing population. Instead of controlling the environment for the benefit of the population, perhaps it's time we control the population to allow the survival of the environment.
And there's another little song that you can listen to later, and then that's it, finished. And it seems all the more poignant now we've got an Englishman up there in space doing spacewalks. And this is a picture my son took last week. He flew off last Tuesday to Africa, and it's his African sunset. So it all seems very much more poignant to me now with those things going on. But anyway, any questions? Happy to take them. Otherwise, you can email me or tweet me or whatever. Nearly kept to time.